Thank you for the organizing committee to bring me here to share my thought with you. So this is my disclosure. So the question is why we need to have a new therapeutic modality. So I think today I brought a couple of challenges that needs to be addressed. The first one is undruggable targets. I'm sorry. So most undruggable targets are intracellular proteins. So we need to deliver chemicals or large proteins or other the therapeutics into the cells. But the protein and RNA therapeutics are not permeable to membrane. So we have to deliver the large keys into cells. The next challenge is disease complexity. Diseases are so complex, meaning that many different cell types, many different cellular pathways are involved. Even the same cell types, they are actually not same. They are quite heterogeneous. And furthermore, these complex diseases are changing. So can we make multifunctional or multi-targeting drugs? More importantly, how can we harness our body's natural ability for self-healing? Our body knows better than we do. So we should address our own body's ability for self-healing. Here's an example. You know, if you think the sparrow ate all grains and fruits, you need to kill all sparrows. But when you did that, what happened? Actually, the population of locusts boomed and causing a terrible great famine. So more than 10 million people died of starvation. On the contrary, if you provide too much, the same thing happens. If you provide too much shrimp to the river or lake, they will eat every zooplankton, which are main food resources for a salmon in the lake. So the number of the salmon decreased dramatically, and it took several years to take them back into lake. So you intervene in a specific pathway, even you don't understand comprehensively, will cause a breakup the homeostasis and end up with exacerbating the disease. So the next challenge is the organelles as a therapy. And this is the levels of organization, starting from atom all the way to the organism, like human. So we may need to pay attention to organelles. So small chemicals corresponding to molecules, protein drugs, antibodies are corresponding macromolecule. Likewise, we need to have a new therapeutic modality at the level of organelle. So can we make a new therapeutic modality at the level of organelle level? The answer is yes, because we have EV. The last challenge is intracellular delivery of key regulators, such as transcription factors. In the couple of decades ago, the Yellowstone, the ecosystem of Yellowstone was destroyed because the number of moose is too high. So they tried to reduce number of moose, but all in vain. So finally, they decided to release 14 gray wolves to the Yellowstone. And after a while, all of a sudden, the ecosystem of the Yellowstone is almost completely recovered, we don't understand what exactly happened there. But we do understand only 14 yellow graves do rescue the ecosystem of Yellowstone. Likewise, Professor Yamanaka tried to introduce 4 different transcription factors to the skin cell to induce the pluripotent stem cells. And it did, it happened. And we don't understand the exact mechanism, how it works. So I brought these 4 questions. I think the answer is yes, because if we have ability to engineer EV to make it as a new therapeutic modality. So what are the advantages of a natural extracellular vesicles or particles? As I said before, the peptide and proteins and nucleic therapeutics, they are very hard to deliver inside the cells. So if we can engineer EV or extracellular particles, we can do that. And there is a lot of other vehicles to deliver these therapeutics into the cells, like a lipid nanoparticle or viral particle or antigen, antibody drug conjugate, etc. But I think the EV has advantage in terms of safety, redosing, hepatic delivery, and cross barriers and intracellular delivery. But still, we have hurdles. So first one is how to load cargos to EV. The location is matter. The cargo is also matter. So how to deliver EVs to a specific tissue and even a specific cells. How to make an endosomal escape. So we need to modulate pharmacokinetics and dynamics, and many other things like a CMC. Still, we have to overcome these barriers. So what I have done in my lab, we developed 3 different platform technology. One is Maxisome, meaning that we can display an interest of protein to the surface of exosome. We also developed a technology to introduce a protein inside of the luminal side of exosome as a free form. And then we developed a fusogenic exosome to escape endosomal pathway. So this is one of the examples. We developed the exosomal EV sorting motif. It is transmembrane motif, and we made several different mutations and finally identify a very specific motif which can sort a protein within the EV or outside the EV. So compared to PTGFRN, it has 10-fold higher sorting ability. Of course, it depends on different types of the proteins. And then here's an example of sulfur alpha, which is a blockade of CD47. CD47 antibody has been tried in clinical study, but unfortunately it failed because of toxicity. But this sulfur alpha EV does not show any toxicity related with antibody-related toxicity. And second example is delivering of transcription factors. So we delivered transcription factor, for example, HIP1 alpha. That will be explained in the next couple of slides. So if you need more information about this project, you may visit a poster will be presented Yeji Lee, I think Friday or I don't know. So the technical goals for non-neoplastic disease is a cleanup and reconstruction by introducing a HIP1 alpha into cells. So we expect the HIP-1 alpha can increase the cleanup the microenvironment by inducing the phagocytosis and prevent further damage by inducing anti-inflammation. And we need to keep blood supply continuously and supply more nutrient and increase the metabolism. So is it really happen in vivo? So we chose a HIP-1 alpha because, as you know, the HIP-1 alpha is involved in many different cellular pathways like perfusion, metabolism, cell growth, etc. And then we pick up one animal model, which is acute liver failure. Oh, it's time's up. So to make long story short, if you can deliver HIP-1 alpha to the animal, acute liver failure animal model, we can rescue the mortality. And I'm sorry, I used too much time. And then the mechanism is done by analyzing the single-cell RNA expression study, showing that the HIP-1 alpha, I will skip all this study, sorry. So HIP-1 alpha, if you believe me, HIP-1 alpha can actually induce a differentiation of monocytes into resolution phase and macrophage, which are dominantly located in the periportal area. So they can help to recovery of damaged liver tissue and also prevent further damage spreading through the central vein area. So conclusion, can we clean microenvironment? The answer is yes. To prevent further damage is yes. And keep blood supply, yes. And increase metabolism, yes. So we can expect the tissue regeneration by simply introducing one transcription factor to the liver. So we have to understand the EV biology, and we have to understand further about the disease complexity, and all these things to be considered for translational focus. So on the horizon, I have good news. We have 142 clinical trials. Of course, some of them are completed because failed. But still, many things are ongoing, and we have one FDA-based drug going to the moving to the phase 3, and the other one is moving to the phase 2 in Europe. So we are expecting to have the first EV-based drug approval from the FDA. This is my paper. Thank you for your attention.